My phone is not okay.
So structure of DNA. See, what is the DNA? What is full form of DNA? Deoxyribonucleic acid. Okay. Deoxyribonucleic acid. What is full form of RNA? Ribonucleic acid. And this is deoxyribose nucleic acid. Okay. So DNA is a long polymer of deoxyribonucleotides. Okay, a DNA is a long polymer of deoxyribonucleotide. Next, length of DNA is equal to number of nucleotides. What does this mean? Length of DNA is equal to numbers of nucleotides.
Like how long the DNA is, that much nucleotides are present.
Yeah, the more longer the DNA, the more number of nucleotides will be present. Usually it is measured in terms of base pairs. Okay, this is measured in terms of base pair because this is one base pair. Okay, that nitrogenous base. So more the number of base pairs, more longer will be the DNA. Okay. This is also the characteristics of an organism. So each organism will have a particular DNA and the length will be same in all the same species. Okay, all species of that organism will have same length of DNA. Example, a bacteriophage known as 5174 PHI. 5174 has 5386 nucleotide. Number of nucleotides. Bacteriophage lambda has 48502 base pairs of nucleotide. Bacteriophage lambda. Then a haploid content of DNA in humans is 3.3 into 10 raised to 9 it is. Okay, not 109, 10 raised to 9 base pair. 3.3 into 10 raised to 9 base pair. It is not 109, 10 raised to 9. And E. coli is 4.6 into 10 raised to 6. Okay, 4.6 into 10 raised to 6. This E. coli is present in your digestive system, which helps to digest the food. Then let's see the next structure, that is structure of DNA. The structure of DNA is made up of nucleotides. What is DNA? DNA is a long polymer of deoxyribonucleotide. So we have to know what is the structure of nucleotide first. A nucleotide has three main components, which is a nitrogenous base, then it has a pentose sugar, pentose sugar, which is ribose in case of RNA and deoxyribose for DNA. Okay, pentose sugar, which is ribose in case of RNA and deoxyribose in case of DNA. Why it is called pentose? Five carbons are there. See, 1, 2, 3, 4, 5. So an example of pentose sugar is ribose here. We have hexose also. Glucose C6H12O6. That will be a hexose. So this is a pentose sugar and a phosphate group. What? C6H12O6. That is glucose. Glucose C6. 6 carbon. So it is a hexose. Okay. A type of hexose. Now here it also has a phosphate. Okay. It also has a phosphate. So it has a nitrogenous base. It has a sugar and a phosphate. Okay. Sugar and a phosphate. Next. There are two types of nitrogenous bases. Nitrogenous bases are of two types. These are the two types. These are the two types. Adenine, guanine, thymine, cytosine and uracil. Now this is one set and this is the other set. What is the difference between this and this?
Sorry.
What is the difference between the up and the down rings?
Those two.
What is the difference between adenine, guanine and thymine, cytosine, uracil?
The down one is only one single.
Only one ring.
Okay.
Whereas here they have
Conjoined.
two rings. Okay. Down one have only single ring, whereas up there are two rings. You can see the two ring. And this is only single ring. So purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. Okay. Purines, adenine and guanine, and pyrimidines, cytosine, uracil, and thymine. Cytosine is common in both DNA and RNA. You will find cytosine common to both DNA and RNA. Okay. From here, we are talking about pyrimidines. This adenine and guanine will be common for both, but we'll talk about pyrimidines. Cytosine is common for both DNA and RNA, whereas thymine, pyrimidines, and purines, pyrimidine, pyrimidine, pyrimidine. Okay. And thymine is present only in DNA, whereas in case of RNA, the thymine is replaced by uracil. This thymine is present in DNA, but in case of RNA, this thymine is replaced by uracil. Okay. The difference between the structure we'll see later. Here also you can tell what is the difference. Okay. This structure and this structure both are same. Just only one difference is there. That we'll see later. Then a nitrogenous base is linked to OH of a first carbon pentose sugar through a N-glycosidic linkage to form a nucleoside. A nitrogenous base is joined to the sugar through which carbon? First carbon. And this nitrogenous base plus sugar forms a nucleoside. We had studied nucleotide, which contains phosphate, sugar, and nitrogenous base. But here that phosphate is not attached yet. It is not present. When it is only nitrogenous base plus sugar, it is called nucleoside. S come first. So don't get confused. This is nucleoside, then T will come. After addition of phosphate, it will turn to nucleotide. Adenosine or deoxyadenosine. This is one nucleoside. When adenine is added to a ribose sugar, it is called as adenosine. When adenine is added to deoxyribose sugar, it is called as deoxyadenine. Similarly, guanosine or deoxyguanosine, we don't say cytosine, we say cytidine or deoxycytidine. Similarly, uridine or deoxyuridine, okay, uridine or deoxyuridine. Okay, not uridine, it will be deoxythymidine. Okay, why there is no deoxyuridine?
Because uracil is present
Only in RNA and thymine is present only in DNA. So we don't have thymidine also, single or deoxyuridine. We have only uridine or deoxyuridine. Now this is adenine. Okay, this is adenine. This is adenosine. See, this is the base. This is the base, adenine. And this is the first carbon, second carbon, third carbon, fourth carbon, and fifth carbon. You all have studied this structure. What do you call this structure as? What chemistry? Bond length structure, no? Okay. So this is the nitrogenous base and this is attached to the first carbon. Now this is ribose sugar. That's why it is adenosine. This is deoxyribose. This is deoxyribose, this part. Okay. And this is the same adenine base. So it is called deoxyriboadenine. What is the difference between ribose and deoxyribose? See the structure. Just see this ribose structure and this deoxy. One oxygen here, if you see at the first, second, third carbon in DNA is removed. Hydrogen is there. Only oxygen is removed. Hydrogen, no, you all just draw this structure. You all don't show hydrogen separately. In that bond, what structure? Bond length structure. You all don't show the hydrogen. You all just show extra groups. Methyl group or some other group. Hydroxyl group. Now this is hydroxyl group. So there is no need to show hydrogen here. That's why it's not written. Only oxygen is removed. Next is how deoxynucleotides are formed. So these were nucleosides. What we studied was nucleoside. Now let's see how this nucleotide is getting converted to nucleotide. When a phosphate group is attached to nucleoside, it will turn to nucleotide. Nucleoside is nitrogenous base and sugar. So to this nitrogenous base and sugar, which is nucleoside, if you add a phosphate group, what you get is a nucleotide.
Nucleoside is sugar and
Nucleoside is S plus B. Nucleoside is S plus B, you can write sugar plus base. And nucleotide is SBN, sugar, base, and phosphate. Okay. So when a phosphate group is attached to a 5' carbon of a nucleoside, 5' carbon, where is nitrogenous base attached?
fifth of carbon.
No, here it is phosphate we are attaching now. Where is nitrogenous base attached?
Yes.
Which carbon nitrogenous base is attached? This is nitrogenous base. It is attached to the first, second, third, fourth, and fifth. This is how you number the carbon. So nitrogenous base is attached to the first carbon, and where the phosphate is trying to attach to the fifth carbon. When a phosphate group is attached to the OH of the fifth carbon, okay, of a nucleoside, OH of the fifth carbon of a nucleoside, okay, this OH, that phosphate will try to join by phosphoester linker. Okay, phosphoester linkage. So that also is important. Which linkage is present between nitrogen and sugar? Between nitrogen and sugar, it is glycosidic linkage. Between N and S, between nitrogenous base and sugar, it is glycosidic bond. Whereas between phosphate and sugar, it is phosphoester linkage. A corresponding nucleotide is formed. If it is attached to adenosine, okay, then that deoxyadenoside will form. Okay, a deoxynucleotide depending upon the type of sugar present. Okay, whether it will be ribonucleotide or deoxyribonucleotide, depending on which sugar is present. If it is ribose, it is ribonucleotide. If it is deoxyribose, it is deoxyribonucleotide is formed. Here you can see. This is till here, it is till here. Okay, till here it was nucleoside. Now to this, what is attaching is this phosphate. Okay, here there was hydrogen. Here there was OH. OH is there. That OH, H is removed, and this phosphoester bond is formed. Phosphoester bond. Okay, so this is the structure of nucleotide. Phosphoester bond.
So for nucleoside, it is
Glycosidic bond. For nucleoside, glycosidic bond. For nucleotide, it is both glycosidic plus phosphoester. Okay. Two nucleotides are linked through 3,5 phosphodiester linkage to form a dinucleotide. If there is two nucleotide joints, they will join through 3,5 phosphodiester bond. Okay, and depending on number of nucleotide join, we will get a polynucleotide. It will be trinucleotide if there are 3 nucleotides joined, and many nucleotides will join to form polynucleotide chain. But where they are joining at 3' 5' phosphate phosphodiester linkage. 3' of one nucleotide and 5' of the other nucleotide. There they will join. Okay, you can see this is one nucleotide. This is one nucleotide. And this is the other nucleotide. This is pyrimidine because there is single This is purine. Okay, pyrimidine and purine. Now here what is happening? This OH and this H, this O, this H, this OH and this H will be given out to form water. And then this phosphate will go and join to this O. Okay. So this is phosphodiester bond. Now can you tell why it is called phosphodiester bond?
Ester group is present.
Yeah, ester group is present in, what is the bond present in single nucleotide? What is the bond present in single nucleotide? Phosphoester. In single nucleotide, it was phosphoester. What is happening in when they are joining? What is happening? They are joining. So this one is having one phosphoester bond. This one will form one phosphoester bond with this phosphate. With this phosphate, this one has already formed a phosphoester bond. Now this phosphate is going to join this and form another phosphoester bond. Another ester bond. That's why it is called as phosphodiester bond. There are two diester bond, two ester bonds. One with the original base, which is already formed. Now this one will go join here and it will form ester bond. Okay? That is why it is called as ester. Okay, ester means CO. In between O is C dash O C.
No.
Uh,
O dash O
dash dash O dash means both the side it is C, no?
Yeah.
Dash O dash, no? At one side it will be C, other side it can be anything. Okay, but between it is O. So here there is this base. This is the one, and this is OP. That's why it is called as ester bond. Similarly, here it is P, O, and the other. So dash O dash. But since there is phosphate, it is called as phosphoester. Got?
Yes.
Since there is phosphate, it is called phosphoester. Dash O dash no is the ester bond. So since here it is phosphate, it is called phosphoester. And there are two phosphoester bond. That's why it is called as phosphodiester bond. Okay. One phosphate, two ester. That's why phosphodiester. Next is, okay, how a polymer is formed. A polymer thus formed has one end a free phosphate moiety, and at the other end it will have a free hydroxyl moiety. Okay, you will see when the structure, you will come to know when we see the structure. At one end of a polymer chain, that is polynucleotide chain, there is free phosphate end, and at the other end there is free hydroxyl end. So free or OH on the third carbon group, which is referred to as 3 end of the polynucleotide chain. So in a polynucleotide chain, there are two ends. One is 5 end and the other one is 3 end. What you have to know is what is present on 5 end and what is present on 3 end. On 5 end, what is present? Phosphate, free phosphate. On 3 end, there is free hydroxyl. 5P they will call 5P 3OH. 5P and 3OH. A backbone of a polynucleotide chain is formed due to this sugar and phosphate. The sugar and phosphate will join to form the backbone. It is just like making a ladder. You take two bamboos. The long bamboos will be because of sugar and phosphate. That long bamboos which we use to make a ladder will be because of sugar and phosphate. And that what you call? Steps or something. Okay. That will be because of the nitrogenous base. Okay. Okay. The nitrogenous base linked to sugar moiety projects from the backbone. Nitrogenous base is projecting out. See here. This is the free fidation. This is your one nucleotide. This is the nitrogenous base. They are projecting outward. This is free fidation and this is free hydroxyl end. Now this free fidation and free fidation and free OH end are free to join more bases or more nucleotide. So here one more nucleotide can come and join. This side also one more nucleotide can come and join. But it will be always the fidation is free and the 3' end is also free, which is this carbon. Okay, let's see where it is. This is 5th carbon, no? Here it was shown slightly wrong. This is 5th carbon. Then this will be 4th. This is 3rd, 2nd, 1st. See, this is 3rd carbon. That's why it is written as 3OH. On 1st carbon, what is attached is the base. On 1st carbon, there will be base. Then this will be your 2nd carbon. This is 3rd carbon, 3OH. This is 4th carbon. And this is your 5th carbon. 5th carbon, phosphate is attached. 3rd carbon, free hydroxyl end. 1st carbon, adenine is, or cytosine in this case, is attached. This is all about the structure of DNA. Okay. Then structure of RNA. What is the difference between DNA and RNA? Now we have studied. First difference, deoxyribose nuclease, deoxyribose sugar in case of DNA, and the other one is ribose sugar in case of RNA. What is the other difference?
Cytosine is common for
Common for both, but in DNA there is thymine, and in RNA it is replaced by uracil. See here, in RNA every nucleotide residue has an additional OH group present at the 2OH in the ribose. So they have additional OH group. What is happened in DNA? This additional OH group is removed. Not OH, what is removed is only O. Additional O is removed. That's why it is called as deoxy. Oxygen is removed. Decrease. D is decrease. OH oxy. Deoxy means one oxygen is removed. Uracil is found at the place of thymine. Uracil is found in the place of thymine. Thymine is nothing but 5-methyl-uracil. The difference between uracil and thymine, there is one methyl group attached at the 5 end of the uracil. That is all the difference. And that's why the name is, another name for thymine is 5-methyl-uracil. So here you can see, this is ribose, this is deoxyribose. Okay. So this is one carbon removed here. Okay. Whereas here it has one one oxygen is removed here. Okay. You can see oxygen, no oxygen here. That's why it is called as deoxy. Whereas here at this carbon there is O present, OH. So at 2nd carbon, remember if for exam they ask at which carbon there is less of oxygen, at 2nd carbon. Which carbon the nitrogen is added, base is added, 1st carbon. On which carbon there is phosphate joining, 5th carbon. Which carbon there is free OH and 3rd carbon. So 4th carbon is not doing anything. Okay. 4th carbon is not doing anything. All work is done by 1st, 2nd, 3rd and 5th. So this is the structure of RNA. Ribose and deoxyribose. Then see what is the difference here. One extra. Structure is same. Only thing, one extra CH3. The structure of this, if you remove this what is circle, if you remove out, it is same structure. Okay. No other difference. Only one, this is added. No change in bonding also, no bonds will remain same. Yeah. Okay. Then last is DNA as an acidic substance present in the nucleus was first identified by Frederick Mischer in 1869. Okay. He named it as nuclein. He called, since it was present in the nucleus, he called it as nuclein. Now, due to technical limitations in isolating such a long polymer intact, it was not able to isolate it intact. You break and they can isolate, but intact, in full form, they were not able to isolate. The elucidation of structure of DNA, the structure of DNA was not studied for very long period of time because there was no instrument also proper. But later what happened? It was only in 1953 that James Watson and Francis Crick, based on X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. Okay, they studied X-ray. They did X-ray study on DNA. That is Watson and Crick. A very simple, famous double helix model for the structure of DNA was given. So this Watson and Crick, they found the structure of DNA, and they were given Nobel Prize during that time to finding the structure of DNA. So this Watson and Crick model is all what we have already learned. Only they are joining the two base pair. We have studied one base pair formation, single base pair. That is, first there will be formation of nucleoside. After that, a phosphate will join to form a nucleotide. Then many nucleotides will form polynucleotide by forming phosphodiester bond. So one polynucleotide is joined to other polynucleotide by hydrogen bond now. Now phosphodiester bond is one, I said no, one ladder. One long bone of the ladder, the other long. We have studied only the long bones. Now, how these long bones are joined was given by Watson and Crick. That we'll study in the next class. We'll stop here.
Yeah.
No.